this is Sample 1, Video 3, the final video on this Sample 1 series. During this observation process, I encountered a variety of problems. The primary, most prevalent one, however, was the preparation of a slide. I find it very difficult to prepare a slide without causing any disturbance. Since I'm utilizing a light microscope, it is recommended that I use a cover slip, especially when going into higher magnifications. But when viewing specific microorganisms, for example the rotifer, by placing a cover slip causes an immense amount of disturbance that will cause the organism to curl up into an unrecognizable ball. In another case, utilizing a cover slip with larger microanimals like this arthropod can cause that microanimal to become stuck between the cover slip and the slide, making it almost impossible to identify. Over the course of me filming these videos on Sample 1, I noticed that my Peranema population began to decrease significantly. I keep my sample in an airtight jar, and I do not add any organic material to it. Once it's closed, I just leave it alone. By the last few slides that I prepared, I only observed 2 or 3 Peranema out of the entire sample. It would make sense that the organisms that rely on live food are the ones who are passing away without the addition of living organic matter. However, organisms like the Stylonychia, who are primary feeders and feed off of dead or decaying organic matter, are the ones that are thriving in this specific sample at the moment. The majority of the Stylonychia I encountered in this sample were too fast for my camera skills, but I did manage to capture this guy. I was also lucky enough to encounter a rotifer. Like I stated before, when utilizing a cover slip, it causes a disturbance in the sample that often leads to rotifers curling up into an undistinguishable ball. But here in this sample, I found one that was active and feeding. I found it difficult to identify this microorganism because rotifers most commonly have a corona, a crown of rotating cilia on their head, and this one does not appear to have that. However, I knew for sure it had a head with a jaw-like structure, a red eye spot, and an appendage resembling a spur, foot, which is used to attach itself to plant life. All of these are characteristics of the rotifer. The phylum Rotifera is divided into 3 groups, the largest being Monogononta, which has about 1,500 species, some without a corona. I then found this photo of the Dicranophorus lutkeni, which has the same head, body, and foot as my rotifer. I looked more into the genus Dicranophorus and found that my organism did most resemble this genus. This rotifer has a hardened mandible that acts as a jaw. It is a prevalent characteristic of this genus. I further compared my rotifer to the D. forcipatus and noted more similarities. The only difference I did note was the 1 spur my organism contained instead of the 2, but then again, these aren't the same species. Like stated previously, the Dicranophorus use their jaws to break dead or decomposing organic matter into small enough particles to fit into its mouth. It has a segmented body that is semi-flexible and covered by a transparent cuticle cover. The foot, or spur, is located at the end of the body and can be segmented in various species. It is used to attach onto objects while the rotifer feeds. The red dot seen on the head is a red eye spot that is a single photoreceptor, which allows it to move towards or away from light. So that concludes my research on this sample. In the future, I won't wait so long to document the organisms that are in that said sample. Once again, I have documented and cited all of my sources and images in the description down below. Make sure to stay tuned for my next video. It's on a sample from a saltwater aquarium.